Hello there. This is another video on the DNA topic. The DNA topic I done at A2, A2 biology. Quite a big topic. Sometimes it's done by schools all as one topic, but what I would do is I would split it into three parts. And I've already done a video for the first part, which would be first part would have looked at the DNA code and it's the role of DNA in protein synthesis. I would also included things in that like mutations and a bit about epigenetics as well. So that's all done in my first topic, DNA and protein synthesis. And I've already made a video on that topic. And if you need access to that video and the notes, then if you get in touch with me on my mobile number here, I get you materials and you can then benefit from the video, since the video essentially deals with all the possible questions, and you need this set of questions, a question booklet in front of you to work through the video. This is the second DNA topic that I do. I kind of split the rest of the DNA topic into two sections. I call this first section DNA technology, and the other section I call gene transfer and gene therapy. And it deserves to be split into two topics because it's such a big section of the A2 course. As I say, many schools will just do it as one topic. I think if they only do it as one topic, maybe neglect to do it in sufficient detail, and there's just too much content to do as one topic. So this topic on DNA technology, to get full benefit, I'm going to go through this topic, and to get full benefit from it, you need a set of my notes here and a set of questions. And if you have a set of notes and questions, you'll also have a set of answers. Essentially what I'm going to do is go through the topic. I'm going to move fairly quickly through the topic, and as I go through each part of the topic, I'm going to stop and do appropriate questions on that particular area. Maybe in the first instance, and something a bit remiss of me, I didn't number the pages maybe in the booklet, so I'm just looking at the pages for this topic, DNA technology, and the pages run from 1 to 17. So if you would just maybe do that first of all, because I'll be referring to page numbers as I go along. Page 1 to 17 are the page numbers on that. This is on the note booklet, page 1 to 17. Now, the thing to say about this topic is that originally the topic DNA technology was done at AS level. This is before, in the old spec, before 2016. It was done at AS level. It was an AS topic. It was actually an AS module 1. In 2016, then we got the new spec, and DNA technology was taken out of module 1 and moved up to module 5. It was replaced by meiosis. Meiosis was taken down from module 5 and brought down into module 1. So DNA technology was lifted from module 1. So my point I'm making here is that much of the material in this topic were items that were tested originally at module 1 and were considered sufficiently easy enough that AS students could have done them. Now they've been lifted up to module 2. Sorry, lifted up to module 5. My second point is that there's a shortage of questions on some of these topics because the specification's only about 4-year-old. Very few questions on many of these topics. So what you'll see there is I have included a lot of questions which originally were in module 1. So a lot of the questions relating to these topics come originally from module 1, simply because there simply wasn't sufficient questions available at A2 because the questions haven't been asked. The other thing I would suggest is that because these areas were done at module 1, they're less likely to come up on A2 questions. So with that in mind, just to say that within this booklet, just running through the topics here, there are only a couple of topics that are genuine A2 topics. In other words, topics that weren't in the old module 1 AS specification, DNA technology. And I just want to tell you what questions those are, and what areas those are quickly. In fact, the only A2 areas I'm turning quickly to page 4 and page 5. Page 4 is genetic screening using microarrays, and page 5 is just a continuation of that. So page 4 and page 5 are A2, it appeared in the new specification, hadn't appeared before. So page 4 and page 5 on genetic screening, and the other materials would be on genome sequencing, and that would be pages 8, 9 and 10. Sorry, 8, 9, 10 and 11, and those are items that are A2 items. All the other pages here in this booklet contain material that was originally done at AS, and therefore may, to my mind, be less likely to be asked as a substantial question. They may make up some small part of a question, but they're less likely to be asked as a substantial part of a question. Nevertheless, they still have to be known. They're on the specification. So things like the polymerase chain reaction, restriction enzymes, things like probes and primers, and things about DNA analysis, genetic fingerprinting. Those are all AS topics originally, but they're on the A2 specifications, so we better know about them. Okay, so I'm going to start moving through this topic fairly quickly. As I go, I'm going to make reference to questions, and I think it might be useful for you to write beside the area. It might be useful to write the question, so when you come back to revise this, you can see what question to go and look at. So, for example, cutting DNA with restriction enzymes, restriction endonuclease, I've represented question 1. So question 1, the question booklet deals with that. So let's see, you need the question booklet to be able to get the full benefit from this video. Okay, so what I've tried to do is bring some sequencing into this topic, some sort of togetherness and sort of an order and a running order for this topic. And you'll see at the start it said this topic deals with various techniques which scientists can use to manipulate and investigate DNA. So largely this booklet is dealing with the sort of basic techniques that scientists use every day when they're dealing with DNA. Things like how to cut DNA, how to stitch it back together again, how to copy it, how to make multiple copies of it, how to read the DNA code, and how to look for patterns within the DNA code, and so on and so forth. So those are basic techniques. So I've summarized them. I've said essentially what I'm going to do in this book is going to look at techniques. It's techniques that cut DNA into sections, or cut out genes, extract a gene from DNA, locating a gene. How do scientists locate genes? So they've got a whole lot of DNA in front of them. How do they locate the piece of DNA that they want? Maybe the piece of DNA that contains the gene. Remember, a gene is just a short section of DNA, just a short length of DNA. Amplification of DNA, that just refers to if you've got a very small amount of DNA, for example, from a crime scene, say a small bit of DNA from a drop of blood, then how do you amplify that? How do you make multiple copies of that DNA so that you have sufficient to do testing on, and so it's sufficient to test maybe more than once. And finally, then the last part, they're dealing with analyzing the base sequence of DNA. So that's looking at genetic fingerprinting and actually being able to decide if two pieces of DNA come from the same person, or if the pieces of DNA come from people who are related, and so on and so forth. Okay, so I'm going to indicate where these topics were originally done, whether they were done at AS, whether they were AS topics or whether they're new A2 topics. So this first topic, in fact, this might be useful at the start before we go any further. At the top of each page here, I wrote either AS or A2. And when AS is written at the top of the page, it means this material was originally done at AS, and therefore it's maybe of lesser importance. So I'm just going to leaf the pages very quickly here and tell you that page 1 says AS, page 2 AS, page 3 AS, page 4 A2. Page 4 is genetic screening using microarrays. Page 5 A2, page 6 AS, page 6 is a polymerase chain reaction, page 7 AS, page 8 A2, which is Genome Sequencing, page 9, A2, page 10, A2, page 11, A2. And then the remainder of this booklet, pages 12, 13, 14, 15, 16 and 17, all contain materials that were originally done at AS level. So you get a feel for whether you're looking at something that was done at AS level or something that's very new to A2. As I say, you could expect to be asked about any of this, but I think the pages of the A2 topics are you'll find that they generate the more difficult and more substantial questions. So we're looking, first thing we're looking at, cutting DNA with restriction enzymes, restriction endonucleases, and they're enzymes that just cut up DNA. And how do they know where to cut the DNA? They cut the DNA because the enzyme can recognize what's called a recognition sequence, which is a sequence of bases along the DNA that the enzyme recognizes. The enzyme cuts at that sequence. It's called a recognition sequence. Usually, there's other terms, recognition sequence, the term I'm going to use. It's a restriction enzyme will cut a length of DNA into many fragments, all of different lengths. This happens because the recognition sequence occurs at many points along the DNA. So if you have a piece of DNA and an enzyme is going to cut that DNA and it cuts it at a particular sequence, the enzyme may find that that particular sequence may occur several times along the length of DNA. And so the enzyme, if you allow the enzyme to cut the DNA, it'll cut it every time it finds its sequence. So for example, if you have a length of DNA, a long length of DNA, and you say that that's the length of DNA, and you see the recognition sequence occurs there, and there, it occurs 5 times the sequence where the enzyme cuts, then you're going to get 6 bits, aren't you? If you cut the DNA in 5 places, that's going to give 6 pieces of DNA, or 6 fragments of DNA, and that's a common question they'll ask you. And the other thing they'll ask you is, how many pieces of DNA will you get? And the other thing they'll ask you is, why do the pieces of DNA that you get, why are they all different lengths? And you say, well, that's because the recognition sequence occurs randomly. It may just occur randomly, so it may occur there, so that's your first length of DNA. Then you may have to go quite a long way to find it again, two pieces, and then you would go a short bit, there's your next length of DNA. Then you may go a long bit again, and again, and so on. So the sequence occurs, and that's the question you should ask. The recognition sequence happens at random points. So you get multiple pieces of DNA, many fragments, and each fragment is different length. Enzymes, restriction enzymes that cut with sticky ends are most useful, and because if you're cutting DNA from 2 different organisms, so you're cutting some human DNA with a restriction enzyme, and you want to attach that human DNA to another DNA, for example, a bacterial DNA, and you remember that from GCSE. If you did that at GCSE, where you could attach human DNA into a plasmid. Plasmid being the bacterial DNA, then it's useful if the two DNAs are cut with the same restriction enzyme that cut with sticky ends, because then you get matching sort of sticky ends that would fit together, and you can see in the diagram there, so you cut them with the same restriction enzyme, so they'll easily attach by their sticky ends. There's one particular enzyme there, page 2, a particular enzyme called EcoR1. These enzymes tend to have strange names, so this one's called EcoR1. They get these names because these enzymes were originally obtained from bacteria, and their name often suggests the bacteria that they come from. So this enzyme comes from a bacteria called E. coli, which is E. coli, and the R stands for restriction enzyme number 1. So this was the first restriction enzyme extracted from E. coli bacteria, and this enzyme cuts at a base sequence GAATTC. So wherever it finds that sequence, G followed by A, followed by A, followed by T, followed by C. So it has to have all of those bases in a line, and it cuts the DNA, and it cuts it with a sticky end. And I've illustrated there that it could cut that piece of DNA on page 2, and it could be useful, that enzyme, because if it cut the DNA down over to the left hand side and cut it to the right hand side on the DNA there, you'll see it in between those 2 cuts, you've got a gene cut. So that enzyme may be able to cut out an intact gene, and a gene just being a short length of DNA. So that enzyme's been useful to cut out that intact gene. Now, typically what happens then is that you allow the enzyme to cut a piece of DNA. It cuts the piece of DNA into many fragments. You'll see down the bottom of the page there, DNA fragments, and they all have sticky ends. One of the fragments contains the gene. Now the question is, how do you identify that fragment? How do you locate that fragment among all the other fragments? And the answer is you use something called a DNA probe. A DNA probe. And you'll see there the DNA probe attached to the gene. And then the pieces of DNA can also then be separated by gel electrophoresis. So I'm going to talk a wee bit about DNA probes over the page here. So, so question one, just about just to mention the question. So question one relates to restriction enzymes there. It's a very straightforward question, and I'll just leave you to do it yourself. It's actually dealing with this enzyme, EcoR1, and there you are, and it asks very straightforward questions. Question two also has elements about the restriction enzymes. Now, moving on then to page 3, locating genes or sections of DNA. Locating simply means being able to pick out one particular piece of DNA from all the other pieces of DNA. Because if you cut up a piece of DNA with restriction enzyme, you've got many fragments, many bits of DNA. You want to locate one particular piece of DNA. Let's say it contains a gene. You want that one located. And then what you're going to use is a DNA probe. And on page 3 there, a DNA probe are used to locate genes. A DNA probe is simply a short, single-stranded piece of DNA, short piece of DNA that scientists make in the laboratory. They join bases together and they make this piece of DNA, and they know its sequence. They make it with a known sequence. And the sequence that they make it with is a sequence of bases that match some sequence within the piece of DNA that they want to locate. You'll see in that case, I've made the DNA probe. It's usually about 20 bases long. I couldn't be bothered drawing 20 bases long, so I've just made it 3 and 4, 7 bases long. But you see the sequence there. And I've said the DNA probe is such that its base sequence is complementary to part of the base sequence of the gene or DNA section that's been searched for. And if you look down the bottom of the page there, you'll see you're trying to locate the gene. You've got all these pieces and you're trying to locate the gene, and you know that you need to know its base sequence. Because if you know its base sequence, then you can make a probe that matches some part of its base sequence. You'll see the DNA probe matches a short section of its base sequence there. And the thing with the DNA probe, and you could put some of this up, maybe highlight that, is the DNA probe gives off radioactivity. So the DNA probe is radioactive, so it releases radioactivity. Whenever it attaches to wherever it attaches, it's then releasing radioactivity. So it's a short, single-stranded piece of DNA, usually about 20 bases long, and it's made radioactive or fluorescent. If you see radioactive, you're okay. And the idea of the radioactivity is that when the probe attaches to the piece of DNA, it's giving off this radioactivity, and the radioactivity can be detected then, usually by a photographic film put close to the pieces of DNA. Anyway, you don't need to go into a lot of detail here, except to say, notice that the DNA probe binds to the gene or the piece of DNA, but the piece of DNA has to be heated to break the hydrogen bonds within the DNA, so that you get 2 separate strands of DNA, because the probe needs to attach to one of the 2 strands by hydrogen bonds. So that's just an interesting point there. Now, that's so far that's all AS stuff, and I've said a relative question there. A question about that. A question you want to look at the DNA probes would be question 3 part D. Yeah, 3-part D asked about that. You can have a look at the 3-part D. It gives 3 marks to describe a DNA probe. And remember, it was an AS question. Right, so now we're moving on. We're now then moving on to page 4. And page 4 deals with a new A2 topic. This topic is on the specification. It came onto the specification in 2016. And it's a modern technological development based around DNA probes. And the term we use here is genetic screening. And the technology is microarray technology, sometimes also called DNA chips, microarrays or DNA chips. And I've said this is a way of analyzing an individual's DNA using many DNA probes at once, so that many different genes can be investigated simultaneously. So it's an application of DNA probes. And in this case, the DNA probes are all positioned on a grid on the surface of a glass slide called a microarray, also known as a DNA chip. So you have this, you'll see down at the bottom of the diagram, you'll see the microarray or DNA chip. It's really about the size of a glass slide you use for looking down when you're using a microscope. And on the surface it's got a grid area. And in the grid area, you've got lots of little hollows, lots of little spots. And in each of the little hollows is located a different DNA probe. And you'll see in that case there's possibly about 100 little spots on that microarray there. It may be as many as 1,000 little spots, and each little spot has got a different DNA probe positioned on it. And also you'll see there it has multiple copies of the DNA probe molecule. So you'll see some examples there just shown to the right. You'll see, you just basically make those out, but the probe in the left hand side is GAGCTACT. That's that probe. Then there's a probe in the next hollow and a probe in the next hollow. And the idea is that each of those probes matches a particular human gene, matches a particular human allele. So let's just give you a very simple example. Let's say if you wanted to find out, if you take something like tongue rolling, which is not the sort of thing this would be used for, but if you take tongue rolling, well, individuals could be big T, or big T, small t, or two small t's. Let's say you wanted to find out if the individual contained a small t allele. Okay, but you could put a probe in, you could put a DNA probe in one of these wee wells that match the small t allele. And small t allele is just a form of the T gene, and you could put a probe that matches, and then you could find out if a person contained a small t allele. And equally, then you can look for lots of other alleles in the person's DNA. And these alleles may be alleles to do with genetic diseases or predisposition to genetic disease. Basically, you're checking out a person's DNA and you're looking to see, do they have this allele, do they have this allele, do they have this allele, and so on. And you can come away then with a lot of information about the person's genetic makeup. So these microarrays, so you'll see at top page 5 then, how is a particular gene or allele detected? So it's quite a complicated sequence of steps there, well, sequence of points which tell you how you go about finding these alleles. So first of all, you start by cutting the person's DNA into lots of pieces, so that you get all the individual genes separated out into separate pieces of DNA, and then you make the genes or the DNA, make them single-stranded, and you label the single-stranded DNA with a fluorescent dye. And then you then place all the DNA fragments from the individual onto the surface of the DNA chip. And if any DNA fragment is complementary to any of the probes in any of the wells on the DNA chip, then that DNA fragment attaches to the probe, and it hybridizes is the word that's sometimes used, or binds to the probe. And in so doing, when it does that, that causes it to drop off its dye molecule. So the dye molecule is released. The dye molecule, whenever it's released, then causes a color change on the grid. And so then any well or any spot on the grid that changes color indicates that the person possesses that allele is present in the human person's, in that person's DNA. And then so you can check out, for example, for faulty alleles. You can look for alleles for genetic diseases that the person maybe hasn't manifested that disease yet. So you may be talking about a child here who hasn't really maybe developed a disease yet, but you can find out beforehand if the child is going to have that disease by looking for that particular allele. And in recent times, a lot of alleles have been discovered to be associated with all sorts of diseases. They don't directly cause disease, but the alleles have been found that are connected to cancer and heart disease and and Parkinson's disease and so on and so forth. All these diseases. Now, that's the basis of it there. Questions relating to that. I'm just looking to see question 7, question 13, question 5, question 7. Yeah. Question 7 originally came from the specimen paper for this specification. It's not a great question, but it's not untypical. Quite a difficult question, so I'll leave you to look at that. Look at question 7. Maybe at some point in the future I'll make a video where I go through all the questions individually for this topic, but at the moment I'm trying to just go through the theory content, and so I'm not going to stop and look in detail at question 7. But question 7 then also relates to the next part then, and so the next part is the next question about how can a microarray be used to detect or measure how much a gene is being expressed in a cell. And questions are relevant here are also question 7 again, and question 13, question 7 and question 13. And if I remember, question 13 is a very recent question. It was asked, I think it was asked in 2019 about this. So just to start then, how can a microarray be used to detect or measure how much a gene is being expressed in a cell? Just going back to the previous topic where I was looking at DNA protein synthesis, we introduced this idea that not all genes get expressed in all cells. In other words, genes are not, so a typical cell, any human cell contains in the region of 21,000 genes. But not all 21,000 of those 21,000 genes are transcribed and translated in any particular cell. So for example, a skin cell doesn't need 21,000 genes transcribed and translated. So only the genes that a cell need to be transcribed and translated are transcribed and translated. And the word we use here for transcribed and translated, we see those genes are expressed. Genes are expressed when they're transcribed and translated. When they're not transcribed and translated, they're said to be not expressed. So you can use this technology to measure how much a gene is being expressed in a cell. So whether it's been expressed at all, but also you can get a measure of the degree of expression. And the way that works, I've sort of run down through it there in very clear points. I've said if a gene or allele is being expressed in a particular cell, then messenger RNA for that gene will be present in the cytoplasm. So for example, if a gene is being expressed in a gene for insulin production is being expressed in a cell, it's going to be in a pancreas cell, because that's the cell that produce insulin. So that gene will be expressed in a pancreas cell. So you should find then messenger RNA for that gene. It should be out in the cytoplasm of the pancreas cell. Okay, so if you take any particular cell and you look in the cytoplasm and you gather up all the messenger RNA molecules that are in the cytoplasm, then you get a good idea of the genes that are being, you get a clear idea of what genes are being expressed. So you look, first of all, you look into the cytoplasm, you extract the messenger RNA from the cytoplasm, and once again, and this is the strange feature, you attach a fluorescent dye molecule to the messenger RNA. So just like you did with the single-stranded DNA, when you're looking for a particular gene, you can attach then a fluorescent dye molecule to the messenger RNA. And then you take the messenger RNA and you flood it over the surface of the microarray, over the DNA probes. The messenger RNA will bind to the complementary DNA probe, releasing its fluorescent dye. And when it releases its fluorescent dye, the dye then shows up as a color on that particular spot. And the way this works is then the more the gene is being expressed in the cell, the more messenger RNA you get in the cytoplasm. The more messenger RNA you get in the cytoplasm, the more messenger RNA is extracted, more messenger RNA is attached to the dye molecule, and then there's more messenger RNA with dye molecules attached to them available, and therefore you get more dye molecule released when the messenger RNA is attached to the DNA probe, to the probe in the microarray. So the more the gene is being expressed, the greater the degree of color you get at that particular spot on the microarray. The depth of color gives a measure of the degree of expression. So I think we'll just have a quick look then at question 13, which is about that. Question 13. Let's go to question 13. So question 13, and this is, I think this was 2019's question. So you can write 2019. It says gene expression can be investigated using microarrays. Researchers investigating gene expression extracted all the messenger RNA from liver cells. Explain why messenger RNA is used to investigate gene expression. Well, basically you're saying if a gene is expressed, then the DNA will have been transcribed into messenger RNA. So you should expect to find messenger RNA for that gene in the cytoplasm. And the more the gene is expressed, the more messenger RNA you should find in the cytoplasm. And then the second part of that basically asked how then are you using a microarray? So this is 3 marks, and this was 219. 3 marks then for saying how do you use a microarray then to investigate the genes that are being expressed, investigate the genes which were being expressed by liver cells. And you basically say you take the messenger RNA and extract it from the cytoplasm, and you add it to the microarray, or you sweep it across the microarray. Messenger RNA will bind to the complementary sequence in a particular well, to bind to the probe in a particular well, and that means then the fluorescent dye, which is present on the messenger RNA, then will be released, and the more color you get, or the more fluorescence, the more color you get, then the more the gene is being expressed, and those are essentially your 4 marks, and that's gene expression. So this is modern day technology whereby people's DNA can be analysed quite quickly to locate what genes or what alleles they have present. The idea of genes and alleles, we all essentially have the same genes, so everybody has got a T gene. It's just we've got different alleles. So, for example, I may be big T, and somebody else is big T, small t, and somebody else is T, small t. So these are just 2 different forms of the gene. So everybody gets two copies of the gene, as it were. So somebody who's big T has the 2 standard copies of the gene. Somebody who's big T, small t has a standard copy plus a slightly different version of the gene. And somebody who's 2 small t has 2 copies of the different version of the gene, the small t. Good. On we go then to the polymerase chain reaction. Now, as I say, this was originally an AS topic, and all the questions in this booklet are all originally AS questions. I scavenged them from AS papers. There has been no modern day question on the polymerase chain reaction. So the polymerase chain reaction is considered to be an easy enough topic to have been done at AS. I'm just referring to the questions that relate to this. This is page 6, are questions 3, question 8, question 10, and question 12. So you can write those 3 at the top of the notebooklet there. 3, 8, 10, and 12. All those questions. And the questions are very useful in the sense that some of them give very good diagrams. The question 3 has a fairly useful diagram. If you can make out the letters in the DNA, it's a wee bit hard to maybe make it out. But question 3 is a good diagram. And so are all the others. You'll see, in fact, that what I have on page 7 there, my diagram, essentially is taken from one of those questions. You can go and look at them. Anyway, so I'm going to go through this very quickly, because, as I say, this is originally asked at AS, and I suspect you wouldn't be expecting a substantial question at A2. This is a technology that allows you to amplify DNA. It's not a word that I like that much, but amplify simply means make multiple copies of the DNA. So if you get a small amount of DNA from a crime scene, for example, and this will allow you to make a large amount of DNA, a large amount of the same DNA, so that you have multiple copies of the DNA, and then you can send one to each crime lab, or you can store one and keep some for later, and so on. Now, it's essentially, it's quite similar to something you learned about in AS, which was DNA replication. If you remember back to AS, you had something called semiconservative replication in AS, where you had the two DNA strands, the DNA molecule were separated from each other by an enzyme called DNA helicase. Hydrogen bonds were broken, and then you had free nucleotides come in alongside the two template strands, and you had another enzyme called DNA polymerase, and it made the nucleotides join together, and that created two DNA strands, two new DNAs, and you said it was semiconservative replication because each of the new DNAs contained a parent strand or an original strand plus newly constructed strands made from free nucleotides. So that was natural. Natural DNA replication, and that happens inside the nucleus of cells, inside the nucleus of cells. Essentially, then this is a modern day scientific version of DNA replication, where scientists have essentially done the same thing, except that they're doing it in the lab. Slightly different technique, slightly different approach, but essentially the same idea. You need to said the following are needed for the process. I'm reading here off page 6. You need the original DNA, you need free DNA nucleotides, you need an enzyme called DNA polymerase, and you need something called DNA primers. That's maybe new. The primers are new. So the original DNA sample is heated to 95 degrees to separate the two DNA strands by breaking hydrogen bonds. Well, there's the difference with the natural process. The natural process used an enzyme called helicase. Here we're just heating the DNA to 95 degrees, breaks the hydrogen bonds, and then you add when you have two DNA strands separated, you add in something called DNA primers. The DNA primers are, so basically if that's the original DNA, and I'll just draw it sideways like that, when you separate the two strands of DNA, there's one strand and there's another strand. The primers are short single strands pieces of DNA that attach at either end of the separated strands. So there's the two separated strands, and you get a primer attached there and a primer attached there. And they're different primers, but basically they're a bit similar to probes. In fact, you could perceive a question where they ask you to compare a DNA probe and a DNA primer. Very similar, both single-stranded pieces of DNA. In this case, about 20 bases long. They attach to DNA molecules. The difference here is these aren't made radioactive, and the other difference is that they are different. You need two probes here because you need a probe. You need two primers. They're called primers, by the way. You need two primers because one has to match this end of the DNA, and the other one has to match the bases on the other end of it. So the two different primers are added, and the sample's now cooled to 55. So you add in the primers and you cool it down again. When you cool it down, the primers then they make hydrogen bonds then with the ends. So they attach by hydrogen bonds when you cool down. Hydrogen bonds remake. Each primer attached by hydrogen bonds. The role of the two primers, a simple role is to keep the two DNA strands apart. They sort of act like wedges stuck in at the end, so they stop the two DNA strands going back together again. Two different primers are needed because the two ends are different. You'll see the top page 7 there. I can illustrate that. There's the two primers. So the next step then is the temperature is now raised to 70 degrees and free nucleotides are added in. And the free nucleotides then are used to construct the other of the two DNA strands. Question 3 is particularly useful for that. There you can see there's the original strands in step 2 there. Step 2, and there's the primers attached. There's one primer TCA attached there, another primer GAC. And those primers keep the two strands apart, while the free nucleotides, and there's the free nucleotides down there, they line up opposite the nucleotides of the template strands. Once again, you need an enzyme to make the nucleotides, the free nucleotides join to each other, and it's the same enzyme as used in the natural process. It's an enzyme called DNA polymerase, or just call it polymerase. However, in this case, as a notable factor, the polymerase used in this case has to operate at a much higher temperature than the polymerase used inside human cells. Human cells, the temperature would be about 37 degrees. And the temperature here for this polymerase is somewhere in the region of 70 degrees. And polymerase is an enzyme. So you need an enzyme that works at 7 degrees. So you're told that the enzyme used in this case is a thermostable enzyme, an enzyme that can resist being denatured. It's actually it's referred to as Taq polymerase, TAQ polymerase. And that's because it comes from bacteria. It's extracted from bacteria that live in very hot springs where the temperatures are very high, boiling water, and so it's referred to as TAQ. TAQ comes, it's described as TAQ polymerase, and that stands for the name of the bacteria. Thermus aquaticus is the name of the bacteria, Thermus aquaticus, and so it's called TAC polymerase. And TAC polymerase moves along and attaches free nucleotides one to the other by phosphodiester bonds. Use of PCR, well, it's obviously used in crime situations where you have got small amounts of DNA and you want to replicate it, or if you use small amounts of DNA, say from fossilized animals, or any other situation where you've got small amounts of DNA and you want to make multiple copies and you want to make a store of DNA, etc. Now, so that was essentially a topic was asked today. Yes, you can see all the questions that relate to it there. I think I've told you that questions 3, 8, 10, and 12 all relate to that, and I don't think any of them are too difficult. Question 10, I'm just looking at there, nothing too difficult. You've got the answer scheme, and you can check that one out. So that was an AS topic. Now, next topic then, genome sequencing. This is a new topic and came on the specification in 2016, the new spec, and it wasn't asked originally in AS, so I'm going to look at it in a wee bit more detail. Genome sequencing starts off with just this idea. I think it was back in 1991 that scientists decided that they were going to analyze all of the human DNA from all 46 chromosomes of the human DNA. They wanted to analyze it from start to finish. What I mean by analyze it, they wanted to know the base sequence. They wanted to be able to write the base sequence almost like a sentence. They wanted to go, the human DNA starts with an A, and then it's thymine, and then it's guanine, A, T, C, G, and just onwards and onwards. I think it took in the region of 10 years before they knew the whole base sequence of the DNA, right from start to finish. And this was called genome sequencing. So often you'll get asked, what does the term genome mean? And the term genome, for 2 marks, it's the complete, and that's a key word, it's the complete DNA sequence of all of an organism's DNA. So you can do this, it's done for humans. Since then, it's been done for lots of other organisms, including things like bacteria, and even for viruses, and recently the COVID virus, its genome has been sequenced many times. The usefulness of that is that they can check if the sequence changes. They can see new variants of the virus where it's like changes or mutations in its sequence. But there's several other reasons for getting the genome sequence of bacteria and viruses and other pathogens and all lots of animals. So, what is genome sequencing then? By the way, the relevant questions here would be question 11. And this topic lends itself well to an essay question. And there is actually the original essay question, question 11. Look at question 11, you will see that it says something along the lines of the question that I have there. I've just essentially said, define the term genome sequencing. Explain the term genome sequencing and describe the potential benefits of genome sequencing and ethical issues raised by the use of technology. So they wanted they're given 12 marks, so you had to define it, you had to talk about the general uses of it, and then you had to talk about something at the end about the ethical consequences of genome sequencing. I'm just looking at the answer scheme then. The answer scheme, so you needed, there was 12 marks, and the answer scheme had something in the region of 16 points, so it was quite tight. But essentially that was it. They want you to look and know the advantages and uses of it, and then discuss one of the consequences of knowing that person's genome, knowing the human genome, and being able to look at that. That means that if you know the general human genome, then you can look at any differences that occur in individuals. Those differences then would be for different alleles that they have for particular genes. So we bit there on page 8, and I'm not going to spend too much time on this because essentially it's a it's a learning thing, and you just have to go through the points. So I've said we have in the region, when they did work out the human genome, all the bases in order, they discovered that, well, in the region of 21,000 genes. So humans have in the region of 21,000 genes. And remember the definition of a gene. A gene was a length of DNA that codes for the base, codes for the sequence of amino acids in a polypeptide. That was the so-called one gene, one polypeptide theory. So it took in the region of 13 years to work it all out. What it now means is that we now know all the genes in humans. We know where they're positioned. I've seen their genetic mapping. Just basically, you've located where all the genes are located. So, for example, you know we've got 23 pairs of chromosomes. So scientists now know where genes for tongue rolling, which chromosome they're on. They're maybe in chromosome 4. Where the gene for cystic fibrosis is at, and chromosome 8, and so on and so forth. So they know the location. They have mapped each chromosome, and they're able to say this particular chromosome carries a certain number of genes. Just a thought there. If we've got 21,000 genes, and essentially we have 46 chromosomes. Well, those 46 chromosomes are matching chromosomes, so essentially we have 23 pairs. Then that works out at the region of 1,000 genes per chromosome. So 1,000 genes per chromosome. And if I say that a chromosome, essentially a chromosome, just to give a very simplistic picture, a chromosome essentially is a long slender structure, like that, and the chromosome essentially has, a very simplistic picture, the DNA is wrapped onto the chromosome. If I take a wee piece of DNA off the end there, that would be a gene. A gene is just a short length of DNA, and then the next gene would be there, and the next gene, and so on. And so basically, we're saying here that a chromosome has enough, the amount of DNA on a chromosome, if you take all that DNA off, it contains in the region of 1,000 genes per chromosome. So it just gives you a ballpark figure. There's 1,000 genes in every chromosome, giving us a total of about 21,000 genes in total. And you'd probably think, what are all those genes for? But they all have a purpose. Okay, biobanks in the bottom of page 8 are just stores. So basically, they're just ways in which this information has been stored on computers, and they've basically got looking at genome differences for different races in different parts of the world, and so on and so forth. But one of the key uses then, and you'll see the way I've done the notes here, basically taken each of these would represent a use or an advantage of the having got sequencing a gene. On page 2 there, sorry, page 9, so item 2, detecting genetic disease. Well, if you know what the standard genome sequence is for a person, then you can look for differences in each individual, and some of those differences will be in particular alleles, will be the presence of particular alleles that give you diseases. So, for example, diseases such as cystic fibrosis, sickle cell anemia, and diabetes, all these sorts of diseases that are genetic in nature. And you can look and you can locate see the presence of the allele in the person's DNA makeup. Then you can say, even if the person's only a child, if the person's a baby at this stage, you can then predict that they're going to grow up and they're going to develop such and such disease. So genetic screening, this idea that you can screen newborn babies, then can be screened for the presence of defective genes or genes that give rise to genetic diseases, such as cystic fibrosis. That then, one usefulness of that is if you can detect that an individual is going to develop a disease in the future. If you screen the baby's DNA, then you can start possibly treating the baby at an early stage. That would allow early treatment. That may possibly, with drugs, that may possibly be able to stop the disease developing in that particular child. The other thing is modern development is the idea that if you can locate a defective gene in a person, such as someone with cystic fibrosis, where they have a defective gene which causes a faulty mucus to be produced in their body and gives rise to a very difficult disease called cystic fibrosis. Then there's a possibility that maybe we could replace the defective gene with the proper gene, with a proper copy of the gene, and that way we could reverse the disease. That's referred to as gene therapy. And I look at gene therapy as a topic itself in the last of these topics in another video. Detecting genetic predisposition to certain disease. Well, in recent times we realized that there are many diseases which we didn't think were genetic. For example, heart attacks and cancer and Parkinson's and various other diseases that we didn't think had a genetic nature. In other words, we thought they were more to do with lifestyle or something else. We now discovered that whilst they're not genetic, the presence of certain alleles in your body, in your genetic makeup, make you more likely to develop those diseases. More or less likely. So, for example, an individual with a gene that makes them more likely to develop cancer might then be more likely, if they smoked, be more likely to develop cancer, whereas a person without that allele might smoke their whole life and not develop cancer. So so you can now look at individuals and note the presence of certain alleles that you associate with certain disease. Most notably, certain genes called BRCA genes have been located in women that make them far more likely to develop breast cancer or ovarian cancer. So they can do early tests in women to check if the presence of these particular genes, I think they're called BRCA, B-R-A-C, BRCA genes, and if these genes these alleles are present, then they can advise the woman that she may in the future develop breast cancer or ovarian cancer, and then so she can then make decisions about when to have her children, or whether maybe to have her, for example, in the case of ovarian cancer, to have her ovaries removed. Once she's decided no longer to have children, that would prevent the development of that cancer. So that's just one example that comes to mind. Creating 3D models of proteins. This is a lesser point, but basically if you know the sequence of all the genes, if you know the base sequence of a gene, then computer software has now been developed whereby you can feed in the sequence of a gene, the base sequence of a gene, and the computer can develop a picture or a model of the protein of that particular gene, the polypeptides and the proteins that that gene would code for. And that allows you then to recognize the shape, for example, of enzymes that are coded for by particular genes. If you knew the shape of an enzyme, say that was produced by a bacteria, say you knew a bacterial genome, that you might know that you'd be able to work out the shape of the enzyme, then you might be able to produce molecules that would inhibit the enzyme. So you may then be able to stop the bacteria growing by adding an inhibitor molecule that matches its enzyme. So these are the sorts of things going to. And one of the most pertinent developments is something called personalized medicine, also called pharmacogenetics. And this is the part of this that has generated a separate question. So I'll just direct you there to question 5. And we picked up this question, I think is about 3-year-old, the pharmacogenetics question. So, pharmacogenetics is also referred to as personalized medicine. And it's a useful question to look at. And basically, the thrust of this is that scientists and doctors are now able to develop medicines and prescribe medicines that are uniquely related to a person's genome, or the presence of alleles in a particular person. Just to give you an illustration of this, if you go back 20 or 30 or 50 years ago, and someone had a heart attack and came into hospital with a heart attack, doctors would give them a standard drug, standard medicine, and a standard dose of that drug. But even back 20 or 30 or 50 years ago, science recognized, or doctors recognized, that people didn't seem to respond to the same dose of the same drug. Some people responded really well to the dose of the drug and got better. Others didn't respond at all, and sometimes the drug made them worse. And then there were some people sort of in the middle that responded reasonably well. So doctors were recognizing that there was something related to each patient that related to their response to a particular drug or medicine. That now falls within this, the most recent developments now fall within this heading, pharmacogenetics. This idea, pharmaco is the sort of drug aspect of it, genetics is the gene aspect of it. It's basically the study, I'm just reading the definition, how the study of genetic differences in humans affects how particular drugs are responded to in the body, metabolized in the body, which in turn affects the effectiveness in the drug, and even to do with its side effects. I've said the different patient responses due to the way in which the drug is broken down or metabolized inside the body by enzymes. For example, different patients respond differently when given the same dose of a drug called codeine. And you'll see question 5 relates to this relates to codeine. And it says, question 5 is saying, individuals, when they're given a particular drug called codeine, they're given a set dose of it. Individuals fall into one of 3 groups. Those who convert the codeine rapidly to morphine, and those who convert codeine to morphine at an appropriate rate, and those who convert the codeine to morphine very slowly or not at all. So what they're saying is that different individuals react to codeine. Now, the thing about codeine is, if you read there on page 10, codeine, when it gets into your body, it's given to you as a drug, it has to be converted into its active form, which is morphine. And that's done with an enzyme. Codeine has to be converted into morphine. Now, everybody's got the enzyme, but some people have a more active form of the enzyme and some people have a less active form of the enzyme. I've given an illustration that the enzyme is coded for by a gene, the gene E, and you have big E and small e. Big E codes for the functional enzyme and small e codes for a non-functional enzyme. Individuals could be big E, small e, or 2 small e's. Individuals who are big E make large amounts of the functional enzyme that converts the codeine into morphine. Big E, small e makes some functional enzyme, and individuals who are 2 small e's, they make the non-functional enzyme. So when you give the standard dose of codeine to each of these three patients with 3 different genotypes, then they respond differently because they have different enzymes present in their body which convert the codeine into morphine. And you see there, I've said, patients who are big E make large amounts of the functional enzyme, and they convert the codeine to morphine very quickly. So quickly, in fact, that they create such high levels of morphine that it's toxic and it may even kill them. So if you give these individuals the standard dose of codeine, they may die simply because they've got the enzyme that converts the codeine too quickly into morphine. There's too much morphine in their body at one stage, and that may kill them. Individuals who are big E, small e, they convert the codeine at an appropriate rate into morphine because they just have a small amount of the functional enzyme, and they respond well to codeine. By the way, codeine is a painkiller. they get good pain relief. But then you have individuals who are 2 small e's, and when you give them the codeine, then they get no effect from it at all, simply because they don't have the functional enzyme that converts the codeine into morphine, so they don't get any pain relief. If you can know a patient's genotype before you give them the drug, if you can know their genotype, and you can do this then through genetic screening, you can find out what their genotype is, then you know how much of the drug to give them. So the individuals who are 2 big E's, you give them a smaller dose of the drug because they convert it very quickly. Individuals who are 2 small E, you have to give them a bigger dose of the drug, or you have to give them a different drug because they don't contain the functional enzyme. So you can see here how knowing a person's genotype in future, and this will be the future of medicine. In recent medicine, we just give everybody the same dose and hope that they get better. Now you'll be the science that the doctors will be wanting to know about the patient's genotype before they prescribe the medicine. They'll know what dose to give or whether that medicine is appropriate. What's the advantage of this? Bottom page 11. The main advantage of this is that you get more effective treatment. Basically, you get people getting pain relief or getting better from the disease, and you're not killing any patients, which is an important point. And also cost saving. If you're given the appropriate doses of medication to patients, then you're not wasting money on medication. So there's less cost to the NHS of wasted drugs. Now, the last part of this, I'd like to going to look at this fairly quickly. This is from now on, all the material from here on, analysis of DNA, it's all done. It's all AS topics. It's all an AS topic, and originally it was done in AS. So I'm going to move through it fairly quickly. A lot of it is kind of a summary. Page 12. A lot of that's a summary of information you may already know about introns and introns and exons and so on, and talking a wee bit about on page 13, talking then about individuals and their DNA. Analysis of DNA, that is essentially the main area that we're looking at here is something called genetic fingerprinting. The fact that you can take an individual's DNA and you can analyze it. You can find out their sequence, and you can look for certain pieces of DNA that act as what you call markers in the individual's DNA. In other words, you don't look at all of their DNA, but you look for certain marker areas of DNA. I've said halfway down page 13, I've said in comparing the DNA of different people, scientists can look for differences in their alleles, that would be their coding DNA, and their non-coding DNA. These areas of difference are known as genetic markers. So if you want to know if two people are related, brother and sister, for example, well, you know that they're going to share a lot of their DNA because they have the same parents, and they should have the same sections of DNA in the same places, and so on and so forth. Two markers that we're looking for, and they're called SNPs and microsatellite repeat sequences. SNPs. Now, I'm downplaying this. Question 4 is the relevant question here. And this used to be asked, bear in mind, this used to be asked at AS. So even though it seems pretty hard and difficult area to understand, because it was asked at AS, you'll see that they didn't ask very difficult questions. So in question 4, you'll see that they ask a bit about restriction enzymes to begin with. And then part C, really, 4C, and they're talking about these two kinds of markers called SNPs, single nucleotide polymorphism. Sometimes they're called SNPs. And the other one's called microsatellite repeat sequences. Microsatellite repeat sequences are areas of your non-coding DNA. Remember, going back to the last topic. Well, if you did the last topic with me, you learned that there was 98% of our DNA is non-coding. In other words, it's in code. It's not genes. It's not part of the code for polypeptides and proteins. It doesn't code for anything. And this non-coding DNA among different individuals, you've got differences in it. Now, one of the differences is the presence of these microsatellite repeat sequences. And they're simply, I think I've illustrated fairly well in the notes there on page 15. Microsatellite repeat sequences are strange repeating sequences in the DNA that run in sequence. So for example, I've used the illustration there that there may be some point in your DNA where the sequence CAT occurs. And you may find this sequence repeats over and over again. So you get CAT, 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 CAT. And it may run for 40 repeats, or 100 repeats, or 5 repeats, or whatever. And what we've discovered is these repeats are present in everybody's DNA. So if I have a repeat that goes CAT in my non-coding DNA, it's present in your DNA as well. But the difference is, if you're not related to me, the number of times it's repeated is different for you than for me. Whereas if I was related to somebody, we would find that me and my relative would have similar numbers of repeats. Two identical twins would have the same number of repeats. So you can determine this. Then microsatellite repeat sequencing used as markers. You can cut them out of an individual's DNA and measure the length of them, get the length of them, and then you can compare that DNA with another DNA. And you can pick on a whole lot of these markers. So within our DNAs, you may have a sequence that goes CAT and it may repeat 10 times. There may be another sequence that goes TAG and it repeats a number of times. Another sequence. And what scientists do is they pick on about 10 of these different sequences, 10 of these different microsatellite repeat sequences throughout your DNA, and they cut each of them out. And the more repeats in a sequence, the longer the piece of DNA is. And that allows them then to match take two persons' DNA and see if they've got matching amounts of repeats of each of the sequences. Now, the only questions that were asked about this was question 4, and you'll see there it said individual 1 and part C, individual 1 and individual 2, the sequence there, AT, 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 AT. And you can see there that you could say that the sequence is a repeat just of two bases, AT, 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 AT. Individual 1 would have 1, 2, 3, 4, 5 repeats, whereas individual 2 has got 1, 2, 3, 4, 5, 6, 7, 8 repeats. And so if you see there, if you cut either end of the repeats, you can see that the piece of DNA in individual 2 is much longer. Yeah, and they're going to ask you what is that. Identify the type marker shown for, for Microarray. Well, they're repeating, and that's a microsatellite repeat sequence in MRS. And then this in marker B. Marker B is a SNP, and all a SNP is just a single base that's different. Single nucleotide polymorphism just means a base that's different, a mutation. And you can see there that marker B is just one base is different. So that's a SNP, SNP, or SNP. Now you can use these microsatellite repeat sequences in DNA fingerprinting. And the process for DNA fingerprinting is quite complicated. You'll see there that the question that relates to this genetic fingerprinting, question that relates is question 12. Now question 12 is an essay question, and I have to tell you that it came originally from an AS paper. So question 12 is an essay question from an AS paper. It's quite a good question because it asks you for 10 marks there. It says, describe how you could amplify small amounts of DNA. So that's, and then it's asking you for the second part, how could you use that piece of DNA then to produce a genetic fingerprint? So basically for 10 marks, they're giving you, they want a summary of the PCR process and genetic fingerprinting. And if you look at the answer scheme for that then for question 10, you have quite a nice wee summary of both processes, both the polymerase chain reaction. This is question 12. You have a nice summary in the mark scheme there. So I'm just looking in the mark scheme. The first 1, 2, 3, 4, 5, 6, 7, 8, 8. About 8 points. I'm giving you about 8 marks for PCR process. There's a nice summary of it there, which hopefully fits with what I have in the notes there. And then the last 1, 2, 3, 4, 5 points. 1, 2, 3, 4, 5, 6 points then are to do with the genetic fingerprinting process. As I say, this was originally asked at AS. This essay was at AS, Module 1. So I'm not going to go through the individual steps for the process. They are there, and you can read through them yourself. The end result of genetic fingerprinting is the sort of pattern you have on page 17 there, the bottom of page 17, where you get these pattern of bars, and each bar represents a different microsatellite repeat sequence. And the position of the bar represents the size of the bar, the length of the microsatellite repeat sequence. And you can see there, and this is an old AS question. By the way, this is from an old AS question. The questions that relate to this are questions 2 and question 6 in the booklet there. So you can have a go at question 2 and question 6. They're pretty straightforward. And question 2 is maybe a bit more difficult. Question 6 is a bit easier. So there you are. That's that half of the DNA topic, and I think you'll agree with me. There's a lot of detail. There's a lot of detail in there. I think the materials in this booklet are maybe less important than the last booklet. The last booklet I'm going to look, or last topic I'm going to look at is gene transfer, which basically looks at transferring genes from one organism to another and gene therapy, and that's a colossally big topic as well. So this topic deserves to be. This booklet deserves itself, as it were. And there's enough information in there to focus on. Okay, so thank you very much, and hopefully you have a slightly better understanding of some of those areas now. Okay, thank you very much. Bye.